and Adrian Gonzalez, who not only are very good researchers, but moreover for me are very good friends. So as it's the last session, so I would just like to say a word to thank all the speakers and participants. Until now, it was very nice to have essentially the same people connecting regularly, so we could have a discussion, and so it was very nice. And so finally, but yeah, we are not sure at the beginning that the format could be. Yeah, it was a big question for us. Would it work or not? That it was not the initial plan to do that online. So as we told you, we will try to organize a workshop session in autumn. So it's not very clear yet how it will be. So we have to think a bit to propose something interesting, and not just yeah, come and let's have fun, but maybe try to find some good subject to think about together. So if anybody has a good idea or examples of similar workshop, and yeah, want to share some ideas, don't hesitate to write to us, and in any case we'll think about how to do something nice in autumn, because hopefully it will be a lot to travel. So let's begin with the talk of Anna. So Anna is in Bonn, and she defended last year it was.
Yeah.
And so in autumn she will join the United States to continue as a postdoc. It was a bit delayed because of this nice period, so it allowed you to stay a bit more in Bonn. So thank you very much for being here, and so I let you present your talk. So thank you.
Thank you, Charlene. In addition to thanking you all for sticking around for the last session, I also, as all the people before me, want to thank all the organizers for their resilience in making this meeting happen, really, finally. This has somewhat become a very nice tradition over the last 4 years, ending the week with these talks and discussion sessions. I'm somewhat lucky to have missed a couple of the talks, so the videos will allow me to continue this for a couple of weeks, probably. Yeah, but it would be even better to see you all in person someday. That would be very nice. All right, so let's begin with the talk. I think this is the title that I promised you, Tumor Heterogeneity as a Cause of Therapy Resistance. And this is an applied project here that we are doing in Bonn together with a couple of people from the medical department. So main collaborators on this project currently are Anton Bouvier, my supervisor, and then Michael Hölzel, and a former student of his, Nicole Glodde, who is now a self-sufficient researcher. Both of them are working in the Institute of Experimental Oncology. And then as it goes with these long-running interdisciplinary projects, there are, of course, tons of other people who were involved in this project. For example, Lorraine, who unfortunately cannot be here today, but she was also part of the beginnings of this project. All right, so the motivation for what I'm going to talk about, there are, so I'm going to talk about cancer, as the title suggests. And when treating cancer, there are a couple of different therapy approaches, and classical treatments that have been here for years and years are treatments like surgical removal of the tumor tissue, radiation, or chemotherapy. And what all of these approaches have in common are that they're rather unspecific, and they're probably good for removing the bulk of the tumor, but usually also harm the surrounding healthy tissue. So surgical removal was pretty clear. Chemotherapy usually also harms cells that are surrounding, and so on. And so the idea of modern therapy approaches is to somewhat derive a more targeted therapy. Those are, for example, small molecule treatments where, as the name suggests, small molecules target some molecular pathways of the tumor cells to inhibit their growth or induce cell death. And then there's a variety of different approaches connected to utilizing the immune system, so the defense system of the body itself. And what these approaches, as the name targeted already suggests, have in common is that they build on a specific marker that distinguishes the tumor cells from the healthy cells. And as this, they are usually a lot less harmful to the general tissue because they only harm the tumor cells. However, that of course means that we first have to identify such a specific marker for the tumor cells, which in general is not very easy. And also the downfall kind of this targeted therapy approaches is that slight variations in the tumor genome or in the phenotype can change this specific marker property and therefore cause a resistance to the therapy, which often ends in a relapse of the tumor. So this is what we want to study in more detail. And of course, we want to use mathematical modeling to better understand the underlying mechanisms and possibly make some suggestions on how to improve therapy protocols. So that's the idea. And with this modeling of tumor heterogeneity come a couple of questions, and there are possibly many more, but these are like the main questions that I'm asking myself. And that is how to describe this heterogeneity mathematically. So there are, for example, the choice between distinguishing just discrete cell types or working on a continuous spectrum. There's the possibility of distinguishing between acquired resistance that only arises under treatment or selection for pre-existing clonal variants of the tumor cells. And then there's, of course, the question of reversibility of this resistance. So is this something that if the treatment goes away, will return to its natural state, or is that a permanent state that is acquired? And when talking about resistance and heterogeneity, one thing that is very important is to have a good notion of fitness, because we somewhat want to describe how these different variants will evolve under treatment or without the treatment. And this needs to be encoded in some kind of fitness or selective advantage in order to know how proportions between these variants will develop. And this depending on the model that you choose, this can be either a fixed value or it can also be seen as a variable quantity. So these are somewhat the questions to keep in mind during the talk. And as an example, we want to study immunotherapy of melanoma, which is a type of skin cancer. And we want to see how we in the past made some of those decisions in this specific example. But I have to tell you, we don't have the ultimate answer. So this is probably also a nice thing to discuss in the end, because there are many different possibilities of how to do it. All right, so the plan for today is that first I want to introduce you to the general underlying model of adaptive dynamics. Most of you probably know about this model. Nevertheless, I want to do a quick recap, also to kind of set the notation, the terms for what's coming afterwards. And then I'm going to tell you a little bit about the experimental background of this melanoma model. afterwards, we're going to discuss how this standard adaptive dynamics model is extended to fit the experiment. And then I will show you, of course, the results of our modeling simulations. And in the end, we're probably coming back to these questions here, and we'll see what we have learned and what is probably still open to discuss. All right, let's get started. So quick recap on adaptive dynamics. So the assumption is we have some trait space, which I used to call V, because we think of this when working in this model theoretically. We think of the trait space as a graph where arrows give you the option of mutation between different traits, and then V stands for the vertices of this graph. And by this quantity N, we denote the number of individuals of a certain trait at a certain time. So that is kind of the state of our population that we want to study. And then we, of course, have to talk about the dynamics. And we're considering so-called individual-based, sometimes also called agent-based Markov processes of adaptive dynamics, where there are different events that occur at exponential rates. So we have exponential clocks associated to different events. And whenever such a clock is triggered, the event occurs and the rates are afterwards probably changed and everything's reset. Yeah, so what can happen in this model? If we're talking about the evolution of a population, we of course have birth and death events in this population at certain rates, B and D. So those are the per individual rates, and then to get the overall rates of such an event, we have to multiply this by the current size of the subpopulation. I should mention that this model assumes that everything is well mixed, and we really don't have to distinguish between individuals of the same trait. And then to make this into an interactive model, so this type of picture we will see later on as well. This is not the trait graph. In these type of pictures, by these circles, I denote different traits, and each arrow corresponds to one of these events with the exponential rates. Okay, so in order to get some interaction, we add a competitive term to the death rate that displays the competitive pressure that individuals of a different or the same trait impose on individuals of trait V. And this is usually put as an additional death rate. One could also think about having this as a birth decreasing factor instead of a death increasing. And since we want to look at evolution over a long time, these types of systems are pretty similar to Lotka-Volterra systems, but we want to look at evolution, so we add mutation to our system. So with a certain probability at a birth event, the birth is not a clonal one, but we have a mutation, and then this M here encodes the law of the mutant. And yeah, so this is our interaction diagram out of the perspective of the trait V. And then these arrows, of course, exist for every single trait. So that is the somewhat known general model of adaptive dynamics. And there's a canonical deterministic equivalent of this model, which you can get by a law of large numbers. That's a pretty classical result by E.T. Kurtz or Fournier-Milliard, depending on whether you're looking at a finite or continuous trait space. So if you rescale this competition kernel, that means less self or inter-trait competition between individuals. And as a result, the equilibrium size of the population increases with K. So if we then rescale the population size by K, this gives us a kind of law of large numbers which leads to this nice differential equation. And for the case of mu being equal to 0, this second term vanishes. And this is also just a 1. So this is then a classical competitive Lotka-Volterra system, just that we have the mutation in here as well. Right. And when working with these models in more theoretical approaches, one usually assumes that there's some existence of a stable equilibrium of the mutation-free system. So for mu equal to 0, we want to have the existence of stable equilibria that could be monomorphic or also coexisting polymorphic equilibria. But in general, there could be many other things happening in this Lotka-Volterra system. For example, if we drop the assumption that this C here needs to be positive, we can construct predator-prey dynamics, and there one really has this cyclic behavior, so no attractive equilibrium. And the last thing that I want to say about the theoretical background is that I mentioned in the beginning that we want to have some notion of fitness of different traits, and many models have a somewhat fixed notion of fitness or selective advantage, but in this model, really the fitness arises intrinsically as a consequence of the rates for birth, death, and competition. Namely, with the fitness, we want to somewhat associate the exponential growth rate of a certain trait. So if we think of a certain subset at equilibrium and a few new individuals of trait W, so some mutants that recently arose, for example, then the growth rate for these very few individuals can be approximated by this following quantity: the birth and the death rate, and the sum over the competition, just as before, just that in this case, we only consider the competitive pressure from these current resident traits that are large and at the equilibrium. And we also assume that those are reasonably close to the equilibrium. And this is what is known as invasion fitness in the context of adaptive dynamics. And it really is the initial exponential growth rate of the subpopulation of a certain trait. And this trait can prevail and fixate in the population precisely when this invasion fitness is positive. And this is all nice and well if we're talking about equilibria when studying biological applications. It's usually not the case that our system is in equilibrium. So as a more general notion, we want to define fitness dependent on the current state of the population, taking into account the competition with all the living traits at a certain time. And this we want to call context-dependent fitness. Right, so I think that is what I wanted to say about the theoretical background. If there are any questions on the general type of model, this would be a good place to ask them. I also forgot to mention, you can of course always interrupt me with questions at any time. And I'm not sure whether I will always have an eye on chat. So if someone realizes that someone else wrote in the chat, please let me know. All right, if there are no intermediate questions, then let's move on to the biology. What we're considering, as I said, is we're studying malignant melanoma. And in this case, this is a model in mice, so not in human yet, but generally this type of therapy is already in clinical studies as well. And the therapy approach that we want to consider is so-called adoptive cell transfer therapy, which is one variant of immunotherapy. So there are also other approaches of immunotherapy where the immune system in general is upregulated in its activity, or there are also tumor vaccines that precondition the body to certain tumor antigens. The idea behind adoptive cell transfer therapy is that in this case, we do know a specific marker of the tumor cells, which is a particular antigen, so a surface protein of the tumor cells. And the idea is to take T cells that are specifically recognizing this antigen, so they have a corresponding receptor on their surface. I should probably also mention that T cells are a particular type of white blood cells, part of the adaptive immune system, and these T cells, when activated, can recognize sick or foreign cells and secrete certain molecules that then induce cell apoptosis. Okay, the idea is that if this tumor established, the mouse must have had either no fitting or too little fitting T cells. So the idea is to take those T cells, expand them ex vivo, activate them, and then re-inject them into the mouse. And so this is the therapy protocol. The idea is to first inject the tumor cells, let them grow until a certain time, and then these T cells are injected into the mouse, and there are also all kinds of other things happening in the tumor in the therapy protocol, which is there is an additional adenovirus that activates the T cells in vivo. There are some accompanying small molecule therapies, some preconditioning and whatnot. And what you can see in experiments is that this approach initially works very well and the tumor shrinks, but then there's often a case where a relapse occurs and the tumor grows again. Yeah, so to go a little more into the detailed setup of these experiments, the first experiments builds on phenotypic heterogeneity of the tumor. And what the experiments showed is that if we have our initial tumor of pigmented melanoma cells, then the immune cells are injected and initially they successfully kill the tumor cells. But while doing this, the tumor cells also secrete some inflammatory cytokines, which is also helpful because the cytokines help to recruit more immune cells to the tissue, for example. But unfortunately, the tumor cells can also sense this cytokine. And in the presence of this inflammatory cytokine, the tumor cells change their phenotype. They can actually de-differentiate to some degree and downregulate the presentation of this antigen, which is for visually effects nicely coupled to a depigmentation of the tumor as this specific surface protein, the specific antigen, is related to the pigmentation of the tumor. So you really see that you start out with a somewhat black brownish tumor, and in the relapse, this tumor is then almost white. Yeah, so this is a phenotypic escape from therapy. And what one can also see is that if the T cells die out or sort of a while after the therapy, the tumor will change back to the differentiated melanoma type. So pigmentation can return to the tumor also. All right, and the second experiment deals with a genetic loss of antigen, so a change in the genotypic level of the cells, where our experimentalists produced some knockout mutants of the wild-type melanoma cells, where the gene for the specific antigen is destroyed. So these cells are, of course, in reality not blue, but for some reason we decided to color them blue in our pictures. These cells are not able to present this specific antigen at all. And then, not very surprisingly, these cells, as they are permanently, well, immune is not a good word, but cannot be targeted by this therapy, they are, of course, enriched in the tumor tissue under therapy. And today we want to focus on the second set of experiments. This first set was nicely studied by a group of people before me. So before I joined this group, so that included Lorraine Coqui, Martina Barr, and Hannah Meyer, who studied this phenotypic switch as an escape mechanism. And now the new set of experiments is concerned with what happens if this really is a non-reversible resistant variant. All right, so let's look a little more. Yeah, so obviously two different sources of heterogeneity here. And so let's look a little more in detail into the results. So what these knockout experiments do is that they take an initial mixed tumor, which partially consists of wild-type cells and partially of knockout cells. This mixed tumor is injected into the mouse, left to grow, either untreated or with this adoptive cell transfer protocol. And then when the tumor reaches a certain size, in this case 10 millimeters, the tumor is taken out and is sequenced to determine the frequency of knockout and wild-type cells at 10 millimeter size. And what we see here is the frequency of knockout cells at 10 millimeters. This dashed line is the initial proportion of knockout cells. And we see that obviously without treatments, these knockout cells seem to have some kind of reduced fitness in comparison to the wild-type cells, as the frequency of knockout cells is decreased in the untreated case. But not very surprisingly, under treatment, the knockout cells are fit with respect to the wild-type cells because they can resist the therapy and are enriched in the tumor. What is interesting, however, is that we have a huge spectrum of data here. So one would have expected that under therapy, the wild type is targeted by the T cells and pretty much goes extinct, and that the knockout type is fully enriched in the tumor tissue, so close to 100% maybe. All right, and to come back to the theoretical model, this really gives us a switch in this context-dependent fitness. Right. Now, I warned you that these pictures with the many arrows would return. This is now a graphical representation of the extended model of adaptive dynamics. So thinking about the first set of experiments, we have the differentiated pigmented tumor cells. We have the dedifferentiated cells that don't have this antigen, that don't present the antigen anymore. As before, we have birth and death rates and competitive interaction. And now, in addition to the original model, we have a switch between the different types that, in contrast to mutation, is not tied to a birth event. And then you might probably wonder why we have dead and really, really dead cells in this model. That is because the data that we get are is the proportion of different cell types in the beginning and in the end of the experiment, but also nicely enough, since we're working with skin cancer that is visible from the outside, we have the size of the tumor over time as data. And this size, however, of course, also is partially made up out of the dead cells that are not degraded yet. So cells that are have recently died or have been killed by the T cells still contribute to the tumor volume for some time before they are cleared from the tissue. So this is somewhat the setup for the untreated case. And then with the treatment, everything becomes much more complicated. The important thing is that we have these immune cells, the T cells, that proliferate in the presence of their target. So they need the stimulation from this antigen to proliferate. They can also kill the differentiated cells, which gives us this additional death rate here. While being active, as I mentioned, the immune cells secrete these cytokines, which induce an additional de-differentiation of the tumor cells, and which also has some negative feedback impact on the activity of the immune system, because the immune system has this somewhat self-regulating mechanism that it downregulates its activity after a while. All right. And then for the second set of experiments, we have to add a sixth type, which are the knockout cells, which can be produced by mutation from the wild type cells, but then have no interaction with the therapy part of the model, as they're not susceptible. And so these two rates you might have seen have these two weird factors here, which I will say a little bit more about in a bit. So this left part of the diagram is somewhat just the adaptive dynamics model from before, with this added phenotypic switch here. And this part here between the differentiated cells and the immune cells is somewhat similar to predator-prey dynamics, which I already mentioned. So the immune cell kills the differentiated cells and through this proliferates itself. Just that we have these two different factors. This 1 minus number of cytokine is this inhibitory feedback that I mentioned. And then this factor we will look at in a little more detail in a bit. So this is a so-called shielding effect that we needed to take into account to explain the data that we saw. So that is the general systemic setup of the model, and you don't have to remember all the details. Just keep this adaptive dynamics plus predator-prey thing in mind. Right, and for simulations of this model, for the purely stochastic system, one can just use a Gillespie algorithm for simulations, but this is really inefficient, of course, in large populations. So what we do is that we make use of this law of large numbers result, and we have a somewhat hybrid algorithm that uses both stochastic and deterministic simulations. Right. And so just as an example of how we arrived at this model structure, I want to look back at this varying fitness of the knockout cells that we saw in the experimental results. And if you remember, we saw that in the untreated case, the knockout cells seem to be somewhat unfit. However, data on growth of wild type and knockout cells separate from each other shows that these two cell types actually have quite a similar growth rate when they're on their own. So as a result, we encoded this negative fitness of the knockout cells in the presence of the wild type in a high competitive pressure that the wild type puts on the knockout cell. And to explain this shielding factor that we have in our model, first of all, this is what the experiments looked like without the shielding factor. I should have probably said, so this is over time the size of the different subpopulations. So the color coding pretty much stays the same. This is the differentiated, dedifferentiated, and the blue is the knockout cells. And this vertical line is when the tumor reaches 10 millimeters diameter. So that is the time where the tumor is sequenced. And you see that already, since the competitive pressure of the wild type has grown too much, the knockout is already decreasing here due to a negative fitness overall. And so this here is what we would expect, actually, which I said before, that under therapy, this knockout should basically fully take over the tumor and reach about 100% proportion of the tumor cells, but that's not what happened. And so we talked to our experimentalists and together came up with this concept of shielding, where we have this additional factor in the T cell rates. And what this basically says is that, well, we need to take into account not just the total number of differentiated cells, but also the composition of the total tumor. Because of course, if we have these differentiated cells just on their own, they can easily be reached by the T cells. But if these differentiated cells are part of a huge tumor that is mainly made up of dedifferentiated and knockout tumor cells, then these differentiated cells cannot be targeted by the T cells quite as easily, because there are many cells that the T cells kind of have to go through and test whether they can target them. And also a large tumor will have a somewhat necrotic core with not a lot of oxygen, which are not very good circumstances for the T cells to invade the tumor. So we are taking into account this proportion of differentiated cells of the whole tumor, and then this is weighed by some exponent alpha, the influence of this. Yeah, this is somehow a very, very simple form of taking some spatial structure of the tumor into account. It's not a spatial model at all, but it somewhat pays respect to this spatial structure that inhibits T cell efficiency. And then this kind of gives us the model structure, but we of course have to parameterize our model somehow. We need to put numbers to all these different letters. And in contrast to yesterday's talks, we are really not doing a lot of statistics here. That is purely due to the fact that we really don't have a whole lot of data. We're working with sample sizes of roughly 10 mice per experimental condition. The experimental results give us evolutionary paths that are all over the place. So our concept is more to find somewhat biologically reasonable parameters in some range, but focus on the more qualitative aspects of modeling. So general effects that are visible across a certain range of parameters and not really quantitative predictions. So just as an example, however, I want to talk about how we picked this alpha here. And for that, we took a look at the frequency of knockout cells in the whole tumor at a certain size for varying parameter alpha. And what we saw is that, interestingly enough, varying the initial size of the tumor seems to have little to no influence on the result. But the size of the tumor at sequencing seems to play quite a big role. So we check for different alpha parameters here and then varied the size of the tumor at sequencing, and we see quite a bit of variation here. And that is easily explained with this picture on the right side, where we see the size of the tumor over time. So we can see this is the time when 10 millimeters are reached. And the blue curve gives you the proportion of knockout cells. And this is initially quite low. And then once the therapy starts, the knockout is, of course, hugely enriched in the tumor, almost reaches 80% in this case. But then, due to this shielding effect, the wild type cells here can recover from their initial decay. And since these wild type cells are originally fitter than the knockout cells, once these wild type cells reach a critical threshold, the fitness of the knockout cells is turned negative again. And then the knockout cells slowly decrease again. And this also results in a decrease of the proportion of knockout cells over time. And so really the time point of sequencing is crucial, and the longer you wait, the longer the percentage of knockout cells should be according to this model. And so we eventually decided for this alpha equal to 4 in this case, because it's nicely situated, gives us this expected value nicely between the 10 and 11 millimeters. And that is a reasonable assumption to vary the size of sequencing, because really what happens is that our experimentalists measure this tumor size every couple of days. And then whenever this critical size of 10 millimeters is surpassed, the tumor is sequenced, but not precisely at 10 millimeters. Right, so with this, I want to come to the results. This is basically just to show you, this is the experiments without knockout cells. And just the interesting thing is that really, as a result of our stochastic deterministic hybrid algorithm, we see here, both in the experiments and the simulation, that there seems to be some threshold for therapy success. And that is due to the effect of this negative feedback of the immune system. So really, if the tumor is too large, the immune system is shut down again before a critical part of the tumor can be eradicated. And due to this hybrid nature of our simulations, we really get close to this threshold with the same initial conditions, both cases of the tumor almost dying out and the tumor going into relapse just to these stochastic fluctuations. But now coming back to this variability in knockout enrichment, which we wanted to study initially, we have seen that some of this variability can be explained by varying the sequencing time. And that seems to be reasonably okay for the untreated case, but in the case of ACT therapy, the experiment still shows a lot more variation than what we can explain through this time point of sequencing. So what we decided to do is to vary the fitness of the knockout cells. So in this case, really the individual fitness, the difference between birth and death rates. And that really helps to explain basically the full spectrum of data. So if this 0.36 is the mean parameter that we estimated, and then varying around this fits quite well with the variation that we see in experiments. And it's actually reasonable to assume that these knockout cells have different fitness, and that is because of the way of how they are generated. So these knockout cells are generated by a CRISPR-Cas method that cuts the genome, and then a certain amount of base pairs are inserted or deleted, but while fixing this strand of DNA. And so we really do have different, genetically different variants of these knockouts in a mixture present for these experiments. And to verify this, our experimental collaborators took single-cell clones from these knockout populations, 3 different ones, and injected them into the mice together with wild type. And we really see that, for example, this variant 1 seems to be much fitter than the other knockout variants, and because it is enriched much more. So that seems to be a plausible assumption that the fitness of these knockout cells really varies significantly. Right, and just the last thing, the nice thing about doing this mathematical modeling is also that we can do things that are not possible in the experiment. So in contrast to studying these mixed initial tumors, the idea of these experiments is, of course, to study the scenario where mutations occur spontaneously, and this antigen is lost due to a spontaneous mutation. And that we can easily model in our simulations, where we just introduce positive mutation rate, and we really see that the pictures look quite similar. So we also have this increase in knockout cells, and then the wild type can still recover, which is initially not quite clear because the wild type might have gone down to a too low level to recover before this smaller knockout population that stems from a single mutant is enriched enough to shield this wild type. And also the enrichment of knockout cells looks quite similar, a little lower, but generally we still get quite a big spectrum here. And one interesting thing again about the stochastic simulations is that we can change this fitness, of course, by varying either the birth or the death rate of the knockout cells. And that really makes a difference, because if we reduce the fitness by increasing the death rate, that results in a lower number of relapses. And that, of course, makes sense, because decreasing the fitness by a higher death rate means higher overall rates of events. And that just increases fluctuations of the stochastic process and therefore makes extinction much more likely. But this aspect can also be taken as a help in designing treatments, because that really means that if we're working on small tumors, pro-apoptotic treatment in contrast to growth impairing treatment should be more preferable, because this increases fluctuations in the tumor size and might increase the probability of actually having the tumor go extinct. All right, and with this, I come to my last slide. So coming back to our initial questions, what we learned is that really in this tumor context, fitness should be viewed as a context-dependent notion and not just parameters changing between treatment and non-treatment, but really we saw this fitness of the knockout depending on the number of wild type cells. So we had this initially negative fitness. Under treatment, the fitness of the knockout became positive, but then even though the treatment was still going on, when the wild type cells recovered, the knockouts again went into a negative fitness. And we saw that this variability of the experimentally measured knockout proportion could partially be explained by random effects like, well, not so much the initial size actually, but the time of sequencing of the tumor. But really, we also had to take into account some heterogeneity of the knockout cells in order to explain the full spectrum and not just stochastic effects in the measurements, for example. And we also saw that having this hybrid model is really a nice thing, because we do see some random effects whenever we're close to somewhat critical points of the system. So where population can die out, or where we have a bifurcation in the underlying deterministic system. And I want to leave with bringing back the questions from the beginning. So in this case, we saw, looking at this discrete cell types between differentiated and dedifferentiated cells, that I didn't show you today, but the previous paper showed that seemed to be a reasonable assumption for this phenotypics, which, however, for the knockout cells, we have now learned that there really should be more of a spectrum of different cells. And we also saw the difference between this acquired resistance of the dedifferentiation that was induced under treatment and the selection for different clones with these knockout variants. We, of course, also had this reversible and non-reversible resistance with the reversible phenotypic switch and the non-reversible genetic mutation. And some additional questions that I'm asking myself now, where maybe you can also be of help, is that really the question is how to distinguish in experimental data between something that is just a stochastic fluctuation and variation that is really due to different cell types. So how can you tell whether we just have different measurements because, yeah, due to stochastic effects, and where are there really different cell types? And also, of course, if we want to move towards a more quantitative model, the question is really how, if we now have these varying parameters between different mice, you see here on the right, we have, under the same experimental condition, hugely different courses of therapy. So how can we best deal with these obviously varying parameters between different mice when we want to fit our model and do some kind of prediction? And also, if we eventually wanted to employ some statistic models to fit our data, thinking about these very variable paths, what would be a good metric to optimize the parameters? Because with these different experimental paths, you cannot just do like a simple mean squared distance method where we want to fit all of these different experimental paths with one simulated path. That wouldn't really make a whole lot of sense. So maybe you have some input on those questions. Otherwise, I want to thank you for your attention, and open for all kinds of questions.
Thank you very much, Anna, for your talk. So does anybody have a question?
I can start, maybe.
Yeah, as you want.
Thank you for your talk, Anna. Can you just tell a bit more about the stochastic deterministic algorithm? So yeah, how so you switch from stochastic to deterministic when the size is
yeah, not precise. So yeah, there are of course all kinds of different methods of how to do these hybrid algorithms. There are also these Taleb methods, there are diffusion approximations and whatnot. What we're currently using is a model where, so we're not distinguishing by small and large populations, but rather by small and so by rare and frequent events. And the frequent events we simulate deterministically, because those should somehow occur quite regularly, but the rare events, like for example mutations, we want to treat stochastically. And then the question is, of course, where do you do the cutoff between small and large rates? We don't have a perfect answer for that. Our somewhat intuition is to scale this this threshold with square root of our carrying capacity, because that is somewhat the fluctuations that you would expect in the stochastic system. And below this threshold, we want to work stochastically so that, for example, extinction due to stochastic fluctuations, we still want to keep those. But that really is a semi-heuristic argument, because this fluctuations again relates to the size and not the rates. So yeah.
So have you tried changing this threshold to see if it has an impact on the probability of extension?
So we're not really studying the probability of the extinction so much, because I don't know, where are these pictures here? So these different ones here are really due to a varying initial size, and that has much more influence on the result here than stochastic fluctuations. So the stochastic fluctuations only really come into play close to this threshold, and these different curves here are due to different initial values, which we assume because of course not all the injected tumor cells survive the initial injection and contribute to the final tumor.
Okay, so the initial sizes here are not the same, because I
no,
thought
so in order to get these different results, so for these two, for this one and this one, they're the same. Those that are very close to the threshold, those are these two cases here. But the other ones, so when the size here at therapy onset varies, that is due to different initial conditions.
Okay, okay.
One could also possibly do slightly different growth rates or so. That would be another possibility.
Okay, thank you.
I have one basic question. It was you showed some picture on the frequency of knocked-out cell without treatment and with treatment. Yeah. And so you have light blue, blue, and gray points. Do you know? What are the experiments?
That is just in the experiment. So this is all polyclonal data, but they have somewhat different polyclonal knockout mixtures. So they did this CRISPR casting on different subsets of cells, and those are the different colors. But blue points, so here really the different green colors are really monoclonal different variants where they selected single clones and expanded them. Here the different colors are still mixtures of different clones.
Okay, thank you. And I have also a question. About this parameter alpha. If I understood properly, you were not very happy with this alpha.
I'm okay with this.
I mean, yeah. Is the point really the inference or the good alpha, or is that maybe it's not the good function to take the ratio to some power?
Yeah, so that is a thing that actually, I can bring up my backup slides. Very good. Not this one, that one. So this is actually something that a master student has been starting to investigate for us. So with some abstract parameter alpha, the question is really how to biologically interpret this weird fraction here. And so the idea that one of our master students started, now she's unfortunately not here anymore, she finished her project and it's not quite done, as always with master theses, is to do a very, very simple spatial approximation where we still look at a well-mixed model, but we have somewhat two categories of tumor cells, the core and the outer layer of the tumor. And then the idea is, instead of having this part here in the T cell rates, we assume that the T cells can only target the outer layer of the tumor. And the core is somewhat protected, and then there's some switch going on between these layers in order to ensure a reasonable constant thickness of the outer layer that should somewhat reflect the ability of the T cells to invade the tumor tissue.
Okay.
And that seems promising and to yield similar results qualitatively with the shielding effect. So that could be a good way to change the model, and instead of this factor here, have the spatial model. However, probably what one also could take away that yes, this somewhat biological model yields similar results than this one. So let's just stick with this weird factor because it's a whole lot less parameters to fit. Because this, of course, gives us double the amount of different cell types. And how would we infer all of these different rates? Because we have no way to distinguish between these two layers experimentally.